I'm going to put a little timer on so that I don't speak for hours and hours, which I'm sure I could. So it's really great for us all to be here. We're really excited about this. So I think we kind of got recruited into this because I met Becky at a career day, and she was the most enthusiastic person there and got us really all excited about the things that you guys are doing. So we thought it'd be a really great opportunity to come and talk to you about the kind of research that we're doing that we're all super excited about. So I thought I'd start off with why I got into doing my PhD. Timer is starting after my little intro. So I chose to do biology at uni because I was always really interested in the nitty-gritty mechanisms of what was going inside a cell. I always thought it was just really fascinating that this tiny little thing had all these complex machineries inside it. So I always found that fascinating. And when I got to the end of my degree, which I did at Durham, I thought that I wanted to carry on in biology because it was something that really interested me, but I wanted to move more towards looking at something kind of health-based. So I was really lucky that I found a project that kind of combined the mechanisms that go on inside a cell with something related to disease. So what my project is looking at is how a cell divides and how this is important to happen correctly to prevent diseases like cancer developing. So it's really important that when a cell divides, we safely divide all the genetic material that's inside it. But what's also really important that hasn't got quite as much attention is all these little machineries, these organelles inside the cell, which also need to be split equally between cells when they divide so that they can function properly. And what my lab has noticed is that one of these machineries, if something goes wrong with how it divides, this stops the cell being able to divide, and all the genetic material inside it becomes more unstable, and this can lead to mutations developing, and is kind of one of the early stages of how cancer develops. So we've got this idea that these machineries inside the cell are really important because they'll form functional cells when the cell divides, but it's also something that can go wrong towards developing a cell into a cancer. So the idea of my project is to understand a little bit more about how this happens. So we want to look at the kind of basic mechanisms that are going on inside a cell that can go wrong and lead to a disease. So the first thing that we're planning on doing is something that I think is really cool. So we're going to label our cell with lots of fluorescent markers so we can see different bits of the machinery that are going on inside it, and then image throughout the whole volume of the cell and do a reconstruction so that we can watch what's happening and get a really good idea of the kind of split of the cell and the mechanisms and the things that are going on. And the kind of more mechanistic, more in-depth thing that we're going to look at is some proteins that might be involved in how these machineries split. So what we want to do for that is some genome editing. So I don't know if you've heard about CRISPR, which is like a really big thing in the news at the moment. It's a really big technique that's coming into the science world. So we're going to use that as a kind of tool to label some specific proteins. So we're going to insert our label into the genome using this genome editing technique, and then do a lot more imaging to see whether we can see kind of these organelles, so the endoplasmic reticulum in particular, being cut and split between cells, because if that goes wrong, then you can lead to the things that I've talked about. So yeah, we are really interested in this specific process, but I really like it because I can see the more long-term implications of it. So yeah, if anyone has any quick questions about anything specific, hopefully I haven't rambled on too much, then yeah, I'd be happy to answer anything.
That was brilliant, thanks Anna. Okay, who's got some questions? I'll take questions from students first, Nick, and then I'll come to you afterwards.
Why are you looking at the endoplasmic reticulum in particular?
Okay, so I think I skipped past that bit because I got a bit carried away, but that's a really, really good question. So the lab that I work in has already looked at the endoplasmic reticulum a little bit, and they've seen that when they disrupt some of the proteins that are involved in the endoplasmic reticulum separating between two cells, they've seen that there's pieces of the endoplasmic reticulum that get stuck. So as the cell, I keep doing this, as the cell divides, so you've got one cell and then it kind of pinches off and divides into two. That little tube between the two cells, you can see the endoplasmic reticulum stuck. And if you you can do a kind of special experiment where you immobilize the endoplasmic reticulum, and when you do that, the whole cell can't divide and it just gets stuck, and it's that's stuck in the middle. Really good question.
He's like, can I ask my question? Yes, you can now ask your question. Have a microphone.
I'm a teacher, so I sometimes tell kids things and I might be wrong, and I often say that, and I think I've heard this somewhere, you might be able to correct me, that the endoplasmic reticulum is sometimes thought of as an extension of the nuclear envelope. Am I completely wrong there? Or
No,
is there...
no. So I guess when you're talking about the endoplasmic reticulum, you're like, oh yeah, this organelle, and then there's the nuclear envelope, and there's like the mitochondria, all these different organelles within the cell that have different jobs, and they're contained by a membrane. So I was like, yeah, they're separate things. Looked into it a bit more. So yes, they are continuous. So actually the nuclear envelope is part of the ER. So the endoplasmic reticulum and the nuclear envelope are continuous with each other, and it's all classed as the endoplasmic reticulum. The nuclear envelope is kind of a subsection of the endoplasmic reticulum
that has to reform,
that's
isn't it,
very specialised, and it reforms from the ER. That kind of lays over the genetic material and reforms the nuclear envelope.
I love how we can all admit that we all are not experts to that level. Great, should we take one more? Yeah.
Hello.
Hi there.
I'm a teacher, I sometimes get things wrong too. You talked about fluorescently labelling bits and pieces in the cell. What does that involve?
So you can do it lots of different ways, but you can use lots of different labels. There's lots of different mechanisms for getting the labels into the cell, which is probably a bit of a longer answer, but essentially we're using a green fluorescent protein, so we're using GFP, and that's a protein from a jellyfish that is used quite a lot as a fluorescent label, and you can introduce it into your cell kind of temporarily attached to a protein that you're interested in, or you can introduce it and have it integrated into the genome, and it will sit there on the protein that you've targeted it to. And so when that protein is made in the cell, it also makes the GFP protein, the fluorescent green label, attached to that protein. And that's what we're going to be doing with the genome editing. We're going to edit in the GFP to the proteins that we're interested in. But if you want a longer answer to that, I'm happy to stick around afterwards.
All right, amazing. I think we could keep asking you questions for the whole 45 minutes.
I'm sure there'll be a little more time.
But we've got to move on to the next one.
Yep.
Huge round of applause. Thank you, Hannah. Okay, next up is Eilidh. Over to you.
I'm going to steal Hannah's idea for doing a timer because I also might waffle on a little bit. But I'm here to talk about the other end of the scale because a lot of these guys are going to talk to you about proteins and cells and molecular, whereas I work on a much bigger scale. I work on whole brain and MRI biology. So the research that I do has actually always been with me since I was in high school, because when I was in high school we would go down to the town centre for our lunch, and when we're standing there and we have our sausage rolls, we can see this old woman. She's cycling up and down the town high street on a tricycle, despite the fact that she's 70, maybe 80 years old. And then it came to me, I was like, why is it that there's such a difference in the success that people have when they get older? Some people are really successful in that they can maintain their thinking abilities and their problem solving or their physical abilities at the rate that someone our age could. But then there are other people who decline gradually, which is very common. And then there are other people who seem to be okay for a very long time and then drift off really quickly. So we know that education, exercise and social engagement can alter how well we do age, but we don't really know why. So that leads me to my research, which is about healthy aging and specifically individual differences in healthy aging. So the World Health Organization said that healthy aging is doing the things we enjoy doing as long as we can. So medical advances mean that we get to live longer, but we are now discovering more problems, and one of those is the increasing rate of dementia. So by 2025, it's expected that there'll be over 1 million people in the UK living with dementia. But the problem is that we can't predict who's going to get dementia, and we can't really follow everyone for their whole lives to work out who might be. So, there's a real problem there, and that it's not really feasible. So the thing that I do is that I am trying to look at what could potentially give us an indicator of who is going to get better aging and who is not. So what I'm going to do is look at oxygen use, because the brain relies really heavily on oxygen. The brain is about 5% of your body weight, but it uses 20% of the oxygen that your whole body uses, which is like a huge imbalance, which shows you just how important it is. So I think that that's really important, because the oxygen use is exactly what the brain uses to make energy. So I'm going to use MRI, which means we can look at brains without, disturbing them. So we can do it in a whole lifestyle. I'm going to use 2 groups of rats, ones that have like a healthy life. They have like new toys and a healthy diet. And then there's going to be standard rats. So we know that the first group is going to age better because of all these lifestyle studies we've done in humans. But what we don't know is how well their energy use is going to change over their life. So that's what my study's about. So hopefully, if I am successful, we can start to work. We could maybe repeat it again in humans and see if this marker is still important in humans, and then we can start to narrow down this big field of who gets better with age and who needs our help. Welcome to take any questions.
Thank you, Eilidh. Wow, so interesting. Right.
So is your research aiming to make it more preventative? So you're trying to spot who's going to have dementia, or are you looking for a cure, or maybe to stop it before it even starts?
So ideally, we want to do it before we get dementia, because at the moment all of our cures are just to stop the disease progressing. What would be ideal is if we could find out what happens first. But the thing that everyone says about Alzheimer's disease is that tau, the protein that accumulates, happens before any of the behavioural symptoms occur. So we don't actually know who's going to get it, and tau is actually really common in aging anyway. So what we're trying to do, pretty much, we're trying to work out of all of the people who it is we should be following from an early age to see if we can get, we can spot them and narrow down our search field a little bit.
Do we have one more? Right. Just over there, please.
Hello.
Hi.
Do you know? So you know that proteins accumulate on the brain, which is thought to cause Alzheimer's and things. Do you know why things like physical exercise and healthy eating can help have a preventative effect?
Yeah, no, we don't. There's a lot of theories going around, and it generally is based around the idea of brain cell activity. Is that maybe there's, repeated use of the brain cells gives it a sort of a reserve. So if something goes wrong, then there's more capacity for it to change and sort of be more flexible. Whereas if it's not used very frequently, then something goes wrong and then it just kind of falls apart. Think of it kind of like a well-tuned bike or something like that. If your chain, is a little bit, it gets caught and it doesn't move to the next gear, it might not be the end of the world. But if your bike is really rusty and doesn't move, then if you could, a small thing could probably cause it much more harm.
Great questions. Thanks. Thank you very much.
No problem. Thank you.
Better move on to Alice next.
Hi, my name is Alice. Bit loud. So I wanted to tell you a bit about my PhD project, which is in the field of stem cells. So these are the cells that we all start off as at the very first stage of our life before we're even born, and that is when we are embryos. Now these cells have 2 very special defining characteristics. So the first one is that they replicate indefinitely, and the second one is that they are pluripotent, which means that they can become any specialized cell type. So that could be a liver cell, a nerve cell, or a muscle cell. Now, it was originally thought that pluripotent stem cells could only be found in embryos. However, just over 10 years ago, researchers demonstrated that a specialized adult cell could be reprogrammed and brought back in time to return to being a stem cell through certain experimental conditions. And these cells were termed induced pluripotent stem cells. Now, they completely revolutionized the cell biology field because it basically means that we now have a source of stem cells for studying diseases and developing new treatments besides embryos. So what I'm doing in my project is looking at the genetic signature that exists in these stem cells when they come for a person. And I'm trying to understand how the genetics affects how the stem cells become specialized cell types. And I'm essentially doing that by recreating some very important stages of embryonic development, but doing it in a dish. And we basically hope that by doing this, we'll be able to develop more personalized treatments for patients. So yeah, that's it. If you have any questions, then feel free.
Thanks, Alice. Great explanation. Do you have any questions? Stem cell related questions. Jumping over there. Lady here.
I'm a teacher as well, and I just had a lesson about this, and one of the questions from the student was, have there been any treatments developed, so in the last 10 years from this induced pluripotent stem cells being used now?
Yeah, so actually in March of this year, there was the first cell replacement therapy using induced pluripotent stem cells. So this actually was in the eye. So the patient had this disease called age-related macular degeneration, which is incredibly common in the older population. And you basically gradually lose your sight, which is awful. So they basically took this patient's skin cells and then reprogrammed them to become induced pluripotent stem cells, and then from there made retinal cells. And when they were transplanted into her eye, they actually managed to halt the degeneration of her eyesight. So, I mean, this is the first one. We're hoping there'll be many more success stories. So, yeah, it's kind of, we're now reaching that stage of the research.
Thanks. There's one just here. Yeah, cheers.
So you were talking about inducing the stem cells. How do you do that? What are the conditions of doing that?
Yep, so it sounds amazingly straightforward. It's actually just 4 transcription factors. So these basically bind to DNA and alter gene expression. So say in your skin cell, you've got all the genes switched on that tell the cell, I am a skin cell, I will perform skin functions. But then you can introduce these transcription factors, which basically switch off the things that say, I'm a skin cell, and switch on the genes that say, I'm a stem cell, I can divide as much as I like, I can become anything I like. So you basically do this in a dish with these transcription factors in solution, and that's where the reprogramming takes place.
Thank you.
Awesome. Thank you very much.
Thank you.
Laura's up next.
Hi, I'm Laura. So kind of following on from the stem cell theme, I'm also going to talk about stem cells, but in a particular context of how we apply them in disease, and particularly in how in our lab we apply them to gut inflammation. So unlike what Alice does, taking the adult stem cells and reprogramming them to become sort of more embryonic stem cells, we actually are interested in the adult stem cells themselves, mainly because they have this really cool property of the anti-inflammatory, which is great for us as a lab because we work on a group of diseases known as inflammatory bowel disease, mainly Crohn's disease and ulcerative colitis. So I don't know if you guys have ever heard of it. There's a couple of few famous people that have Crohn's disease and ulcerative colitis that you might recognise. Dynamo the Magician, Darren Fletcher used to play for Manchester United. They all suffer from Crohn's disease, and it's a very debilitating disease. It causes a lot of abdominal pain and discomfort, and really patients don't have a great quality of life, and there's currently no cure for it. And the drugs that we do have, while some are great and they work fantastically in some people, there's a lot of people that they don't work in. So we started to look towards stem cells as a new therapy because of this anti-inflammatory property that they have. And what we're mainly focusing on is understanding the mechanisms behind how they work, because whilst they have been taken into all these trials and they've been shown to make patients better, no one has ever bothered to look, why is it that they're doing that? And a lot of patients, again, like the drugs we have, don't respond to stem cells sometimes. And if we don't understand exactly how they're working, we can't begin to answer any questions as to why people aren't responding to them, or even how can we predict whether people will respond to them in the first place. So, yeah, that's just a very brief kind of overview of what I do, I suppose. We're at very early stages of it, but hopefully the goal of this is, at the end of my PhD, once we actually understand all the mechanisms behind it and we can understand how we can make therapy better, we could hopefully feasibly take it into patients in the future.
Cool, thanks, Ola. Hand straight up at the back. Nice one.
Hi, I have heard that controlling your diet can help colitis. Do you think this is true, or in some way?
It can in some cases. I'd say probably the evidence is quite patchy and probably not necessarily well done, the science behind it. But a lot of patients do find that if they restrict their diet, it does help. Some patients eliminate gluten, for example, and find that that might improve symptoms. But it is in no means that is probably never the only course of treatment that patient is recommended for management of the disease.
Take this one here, thanks.
How might stem cells actually be used in a therapy that was going to treat ulcerative colitis, for example?
That's a good question, yeah. So at the moment, we try to focus it to patients who are failing all other conventional therapies that they're given beforehand, because as you can imagine, the process of taking stem cells and growing them and then trying to infuse them back into a patient is quite long. It takes a lot of manpower, it's very labour intensive, and therefore ultimately ends up being quite expensive. So whilst personalised medicine is the way forward, it's still very early stages because not a lot of people are doing it and it's very expensive. But I think as soon as you see a bigger uptake of it, it will fly as soon as it just needs to get into the clinic and into patients to be able to do that.
Cool, and there was one more at the front. Yeah.
You were saying that the drugs that you used on people, in some cases they worked, and in some cases they had no effect at all. Why would the drugs have no effect in some people, but then work on others?
Because the immune system's very smart, especially in the gut, in that inflammation is never caused by a single pathway. There's always multiple pathways that converge to create disease, and the problem is a lot of the drugs focus in on either stopping or blocking one of those pathways. So what we're hoping with the stem cells is because the mechanisms which they use are probably a lot more broad, they've got the chance to block and inhibit a lot more of these pathways at once than just a single drug.
Super interesting. Nick, do you want to squeeze one in?
Were we on the bus from Sheffield? We were really interested looking at your talk title, and I don't think anyone's brave enough to ask the question, so I will. So we're going to talk after you guys about the human whipworm project, and we get our DNA from a Danish scientist who's got an inflammatory gut problem. Okay. So he's treated himself by infecting himself with intestinal parasitic worms.
Braver than I.
Yes. I think his theory behind that is the human gut's grown up with parasites, and therefore the immune system's always busy in the gut. But in the Western world, our immune system doesn't have much to deal with inside our intestines. What are your thoughts on that? I'm not suggesting that you would use that as a treatment.
No, it is actually quite interesting because when the parasitic infections induce a very different immune response to that you see when people have inflammatory bowel disease. And the idea is that if you essentially have a parasitic worm and someone has inflammatory bowel disease, it skews the immune response to something that's more anti-inflammatory rather than inflammatory, which is why there's the suggestion that it may. But I wouldn't recommend trying it myself personally. Very brave person to try it on himself.
Brilliant. Thank you very much, Laura. Okay, Maia's with us now.
Okay, so my PhD project is more about cancer, specifically looking at lung cancer and the role of the immune system. So I'm actually not an immunologist. I am interested in looking at cell migration and cell adhesion, and there's a particular protein that we're interested in studying in our lab, which has a really long name and isn't that important. But we found that if you upregulate this protein, lung cancer cells grow a lot faster. And we're interested in knowing why that is. So how does this protein have that effect on tumours? And some other data that some other lab found out is that the protein I'm interested in is also involved in inflammation in lung cancer. And lung cancer is one of the most debilitating types of cancer. So this is really important research, especially because a lot of the treatments for lung cancer are immunotherapies, which you've probably heard about in the news. It's like a really big buzzword. And they are, when they were first discovered, people thought they were like a miracle treatment, because they were really, really effective of reducing tumour growth, but patients had a really high relapse rate. And this was mostly to do with genetic mutations and things like that. But it's really important that the conclusion was that people didn't really understand how these immunotherapy drugs were working. And because they had such a great response rate in the clinic, people just raced forward, and now there's loads of patients who were relapsing and weren't able to be treated. So the experiments I'm going to be doing are going to be using mice, and these mice are completely normal mice. They're completely wild type. And I'm going to be doing this because a lot of cancer immunotherapy trials started off in mice which didn't have a functioning immune system. And this is done because we have to use human cells in order to study lung cancer. And if you use human cells in regular mice, the mice will reject them. It's like organ rejection. So I'm going to be trying to do everything in a mouse system. And we're thinking that my protein is involved in the migration of immune cells in and out of the tumour. And this, if you have an increased infiltration or more movement of immune cells into a tumour, you're more likely to have less tumour growth, and this is because the immune system is able to recognise the cancer. But if you've got a protein that's upregulated and isn't allowing movement of the immune cells, then you're going to have higher tumour growth. So I haven't actually done very much yet because I just started, but I've just started to work with mice, and it does appear that I'm finding the same results as we found in human cells. So when my protein is at higher levels, we have a lower at lower levels we have a lower tumour growth. So I'm going to start working on looking at the immune system. So in the future, we hope that we could use things like gene therapy, which are also really need a lot more work on, in conjunction with immunotherapy, so that the genetics aren't necessarily as problematic in terms of these drugs causing relapse. If you have any questions, then...
Thanks, Maia. Great, question from the back.
Hi, yeah, so kind of when I saw your title, my eyes kind of lit up, because the other day I actually read an article about the kind of regulatory T cells when they approach a tumour. And what this article was saying was that the whole point of why the immune system doesn't normally respond to a tumour is because it essentially recognises the tumour cells as its own. Is that, so are you kind of contradicting that, or is that what you were saying? Sorry?
No, so I'm not actually sure which immune cells my protein is involved in controlling, but that is essentially what happens. Like regulatory T cells will recognise, that's like another part of cancer immunology, but they will recognise that the tumour is foreign or bad, and will tell the immune system to remove those cells. It's kind of like that's happening in everyone constantly, because the body is constantly misreplicating DNA, and so, but the immune system is usually able to fight that. So it's kind of like using the body's ability to recognise the fact that a cancer cell is there, and that could be regulatory T cells. That's what cancer immunotherapy is trying to do. So it's trying to use the body's own immune system, if that makes sense.
Cool, thanks for the question. Do we have another one for Maia? Sure.
Hi there. You mentioned the penetration of the immune cells.
Infiltration.
Or infiltration.
Yeah.
Is that presumably an increased vascularization of the tumour? Or,
not necessarily. So immune cells are able to exit the blood vessels and infiltrate tumours if they're close enough to blood vessels. Eventually, as the tumour gets larger, then vascularization needs to happen. But initially, I'm just going to be looking at small tumours so that the immune cells can move in and out without needing to have its own like blood vessels. But yeah, that is the thing.
Is there a signal from the tumour that assists, improves this penetration? Whatever word you used. Is there like HIF, for example? Sort of increases vascularization. Is there another factor that the tumour can secrete?
Off the top of my head, I can't think of any. I'm sure there are. Do you guys know of any? So there will be, things like regulatory T cells, that's their responsibility, is to regulate T cells entering those sort of areas. And they use things like cytokines. So that would be a sort of signal that the cell uses to increase like chemotaxis, or so like chemical attraction, essentially, of different immune cells to a tumour area. So there's loads of different ones.
Sounds like it's early days, and it's going to be an exciting, exciting few years. Thanks very much, Maia.
Thank you.
Huzefa's up next.
Hi, everyone. So my PhD is in the neuroscientific research area. So as Eilidh mentioned before, she's looking at the more large-scale brain in Alzheimer's disease, but I'm going to take it down to the other end on the molecular side. So I just want you to imagine one day you wake up and you forget one of your most precious memories, either with friends or family, or you're going somewhere and you forget why you're going there or where you're going. And that's exactly what it's like for a patient suffering from Alzheimer's disease on a daily basis. Now, these precious memories are actually formed from the connection of your neurons called synapses. And essentially, the stronger the memory you have, the stronger the connection. But in Alzheimer's disease, these connections weaken, they deteriorate, and become dysfunctional. And those memories start to fade away. Now, my research is looking to understand the synaptic changes and the mechanisms behind why it might occur. Now, if you take a brain of a patient with Alzheimer's disease after death, what you find is that there's a buildup of lots of different proteins. One in particular that I'm interested in is tau. Now, tau normally is involved in the growth and stability of neurons, but in Alzheimer's disease, it sort of clumps together. It accumulates and it spreads very rapidly. And we think that it's involved in the synaptic changes that occur in Alzheimer's disease. Now, what we found is that the hippocampus, which is a region, or the memory region, is that lots of different synaptic proteins are reduced. And this is one of the first regions affected in Alzheimer's disease, where you see a lot of tau. But in the cerebellum, which is in the back of the brain, which is involved for movement, you don't really find much tau. But these proteins, these synaptic proteins, are increased. And as you can see from Alzheimer's patients, they don't really show any problems with their movement or balance. So we think that perhaps tau is involved in these synaptic changes. So we're trying to look at post-mortem tissues from patients after death, looking at tissues, staining them for different synaptic proteins and seeing whether they relate to tau. Now, the coolest thing about our project is we're actually trying to grow neurons in a dish and trying to model Alzheimer's disease research. Not Alzheimer's disease in particular, but maybe a part of it, such as tau release, and trying to stimulate neurons and seeing the changes that might be occurring within synapses as well. And so we hope that one day we'll be able to try and find a particular protein that might actually protect synapses, and eventually we hope that there will be a treatment for the future. So, thank you.
Amazing. Thanks so much. Great presentation as well. Any questions? Yeah, one just there, please.
So you said that tau tends to build up more in the hippocampus memory area rather than the cerebellum. Do you know why that is?
No, but that's something that I'm looking into. Like, we think that there's certain proteins that may be involved in the clearance of tau, for example. But when tau builds up to, say, a particular level where these clearing proteins or chaperones, they might be dysfunctional and they might not work properly, so tau starts to build. But in other regions, which are affected quite late in Alzheimer's disease, we don't know what's happening with these chaperones at all. And we're trying to see whether these might maybe upregulate or increased and are able to clear tau much better. So, yeah.
Cool, good question. Yeah, I'll come to you.
What made you investigate tau rather than any other protein?
So my lab was sort of focused on tau. There is also amyloid beta. I mean, there's a whole cascade. No one really knows where to begin or where to start from. But I think choosing somewhere and that's your focus is the best way to go forward with that. I enjoy working with tau because you get really pretty pictures. And but other than that, I think there's got to be a really good science behind it. And I really found it really interesting of where it was going. And that's why I sort of followed on with it.
Cool, there you go. Cutting edge research or pretty pictures. It's up to you. Thanks so much.
Thank you.
Martyna, right? Excellent. Last one, sadly. Over to you.
Yeah. Right. 1 second. Okay. So, hi. I am Martyna, and I am actually just starting my PhD. And the first year of my PhD is I'm doing 3 different projects in 3 different labs. So I'm jumping around a lot, and I'm doing stuff with autism and depression on mice and on humans and all that stuff. So I thought maybe I should go back to like a project I actually finished. Because I could tell you more about that than what I'm just starting. And I will tell you about my undergraduate project in my final year, during which I spent 3 months cutting up dead flies, which sounds a bit gross, but it was actually really interesting because we used the flies to find possible targets for genetic therapy in a very rare lung disease. It's a disease that's called primary ciliary dyskinesia. And when you have it, from birth you suffer from very frequent lung infections. Also, you can be infertile, and interestingly, you can even have your heart on the wrong side of your body. And the cool part about it is that it's caused by little hairs on your cells called cilia that you have in your lungs. And when those cilia don't move because of a genetic defect, that's where primary ciliary dyskinesia occurs. And interestingly, this cilia structure is very well conserved in fruit flies. So basically, they have the exact same genes, except for them it's not in lungs or anything, but it is in their auditory neurons. So the genes that would cause primary ciliary dyskinesia in humans cause deafness in fruit flies. So basically what we could do is we could take those developing fruit flies, so fruit fly larvae, and then develop some fruit fly strains, so mutants that are deaf, and look at what exactly failed to cause them to be deaf, like in terms of the genetic protein pathway. And day to day, my lab work looked like I would walk into the fly room, which is a smallish room with lots of microscopes and like shelves of vials upon vials upon vials of like little tiny fruit fly colonies. So it would be like hundreds of thousands of fruit flies in one room. Then I would take my larvae that appeared during the night, and I would dissect them, cut off their antenna, and then label them with a specific antibody, which would cause my protein of interest to glow when I shine a certain kind of light on it under a microscope. And then I could look at how this protein moved during development and hopefully work out what causes primary ciliary dyskinesia and how we could possibly treat it in the future. All right, thank you very much.
Thank you. Right, any questions? About using flies especially. Cool, here you go.
All right, might sound like a silly question, but how do you know if a fly is deaf?
No, this is actually a good question. So you can totally do different behavioral tests on fruit flies. And there's two different types of tests you do. So when it's a larvae, larvae can also hear in fruit flies, and they get scared when they hear something. So what we did is we built a little speaker, and we placed the fruit fly larvae on the top of the speaker. Well, not right on the top, but sort of on a shelf where the speaker was underneath. And when they heard a loud, low-pitched sound, they would get scared and sort of curl up on themselves. Yeah, so that's how we tested it.
Cool. Any more? Guess what? You got a question. Yes, man.
So human hearings, you've
Yeah.
got cilia in your cochlea or whatever.
Yeah.
Is that a kind of parallel evolution, or is that a linkage between them?
Right. Actually,
going back in
yeah,
evolutionary time?
that's a very good question. And the thing is that the cilia itself is very conserved. Basically, even like single cells have them, but there's different types of cilia. And the type that we have in our lungs is the kind that is motile, so it moves and pushes mucus out of our lungs. And in fruit flies, it moves to amplify the sounds. Whereas we also have little hairs in our cochlea, like you said, but they are non-motile. So the only thing that they do is just sort of sway, and we perceive this swaying. So we can't really apply it to human hearing, but weirdly enough, we can apply it to lungs. Which is, it's a good example how you can use sort of like a really basic science topic to something that's seemingly not very connected, but relevant to human disease. So.
Awesome. Thank you very much.
Thank you.
Right, I've got 2 minutes until we need to move on. Does anyone have any questions about the general next steps of the career? You know, what the whole life is like as a PhD student. Can we take the one at the back corner there, and then the one in the middle here?
Hello. I was just wondering, how did you go about, having finished your degree or your master's, how on earth did you go about choosing these wonderful topics that you've just spoken to us about?
Who would like to lead on the trickiness of that? Please do.
So a lot of us, besides Eilidh, did a program where you do rotations in your first year, which is what Martyna touched upon in her talk. So that first year is a Masters of Research, and you do rotations through 3 different labs. We actually had about 80 projects to choose from, spanning cell and molecular biology, which we've talked about today, but also things within psychology and psychiatry. And that was just a really good opportunity to try different subjects which you hadn't looked at before, learn different techniques, and also get a feel for the lab environment that you could potentially be doing your PhD in. So if you are slightly undecided about which direction you want to go in your research, that is a really good way to kind of try before you buy, and also just to develop different skills which you can then go on and use in your actual PhD project. So that was a really, really useful experience for me personally.
Cool. Thanks, Alice. That's it. Cool.
If I'm looking to do a placement year for neurology, what would you recommend doing?
Who's going to jump in that one? Was it neurology or virology? Neurology.
My advice would be have a quick look on Google Scholar, see if there's any particular fields in neurology that you're interested in. I think these guys are all really surprised all the time that neuroscience is so broad, that we don't actually understand each other's research. So work out if there's a general disease you'd like to work on, or if there's like a technique you'd like to do, and then find someone who does it really well, and then send 1 million emails. Just like bug everyone. Just relentless emails will get you somewhere, and then but 100% once you get your foot in the door and get some practice in someone's lab, then it's not quite a cakewalk, but like it's a lot easier to move forward from there.
Great question, great answer. Can we squeeze one more in for this young man at the front?
So I was wondering, as a PhD student, do you get some sort of income, or do you have to fund your life?
Talk about the money.
So different PhDs and different programs that you can do a PhD in have different setups. It is an option to fund your own PhD, but all of us are funded PhD students, so it's possible to apply either for a program which has funding attached to it, which is what we're all doing, or you can apply for a PhD project specifically that you're interested in, and then apply to a funding body to support that research. Sometimes labs say, I've got funding and I've got a project, are you interested? So there's kind of a spectrum of options depending on what you're interested in and depending on where you're applying at the time, but there are lots of funded opportunities.
So you sort of get less choice on what PhD you want to do if you want to get it funded, I guess.
Not necessarily. Like, for instance, our program is pretty broad, so there's a wide spectrum of options, and it's fully funded. There are other projects where some people know exactly what they want to do and they know who they want to do it with, so they're just kind of waiting for, you know, a project to come up. And there are people who just know what they want to do, and then they go to the supervisor themselves and say, I have this great idea, you're the person who can take me there, let's apply for funding together. So, you know, there's a couple of options.
Great question. Right, sadly, we need to push on, but that was brilliant. Thank you, Hannah, for bringing everyone along and setting that up. That was really, really interesting.